Today, Tuesday the 25th of September 2018, TIDMSCLP RNS Number 7739B Scancell Holdings Plc. The 25th of September 2018, the 25th of September 2018, Scancell Holdings Plc, Scancell, or the company. Final results for the year ended the 30th of April. 2018 new collaborations provide strong validation of immunotherapy platforms. Scancell Holdings Plc, the developer of novel immunotherapies for the treatment of cancer, today announces its results for the year ended the 30th of April 2018. Highlights: corporate, £11.6 million net proceeds raised in a placing of shares of funds used to initiate the clinical development of SCIB1 and to continue to support the immunotherapies platform pipeline. Dr. Cliff Holloway appointed as Chief Executive Officer, succeeding Dr. Richard Goodfellow. Agreement with the University of Nottingham to license several novel monoclonal antibodies against tumor-associated glycans. Immunobody. Impressive 5-year survival data from the SCIB1 Phase 1/2 clinical trial. All 14/16 resected stage 3 IV melanoma patients receiving 2 to 4 mg doses and who remained alive at the end of the study in March 2018 reached their 5-year post-treatment survival time. Proposed international SCIB1 and checkpoint inhibitor combination trial planned to commence by end of 2018, subject to necessary regulatory approvals. Peer-reviewed research paper on SCIB1 Phase 1/2 clinical trial published in the scientific journal Oncommunology. Clinical development partnership with Cancer Research UK to develop SCIB2 for the treatment of patients with solid tumors, including non-small cell lung cancer. Patent granted in Europe for Scancell's DNA immunobody technology. Counterparts to this patent have already been granted in the US, Australia and Japan. Immunotherapies first-in-man clinical studies in MODY1 for triple-negative breast cancer, ovarian cancer and sarcoma anticipated to commence in 2019. Collaboration agreement entered into with ESA Pharmaceuticals for the manufacturing, development and commercialization of MODY1, AMPLIVANT conjugates. The polypeptide group contracted to manufacture clinical supplies of MODY1, AMPLIVANT conjugates. Collaboration with BioNTech to investigate T-cell receptor-based therapeutics for the treatment of cancer, expanding potential therapeutic utility of immunotherapies to adoptive T-cell therapy. Scancell, as part of an international, multidisciplinary team, shortlisted to the final stages of Cancer Research UK's Grand Challenge in Cancer Vaccinology. Financial loss for year of £4.2 million 2017, loss £3.5 million Group cash balance at the 30th of April 2018 was £10.3 million the 30th of April 2017, £2.7 million Post-period highlights: broad patent granted for Scancell's immunotherapies technology, covering all major European territories. Strategic research collaboration with the Rheumatology Unit at the Karolinska Institute expanded to explore the potential of immunotherapies to develop multiple immunotherapeutic agents for a range of cancers. Scancell exercised its option to a worldwide commercial license for the use of Ichor's Trigrid 2.0 electroporation delivery system with SCIB1, an additional £1.1 million net of expenses raised in an open offer to shareholders in May 2018. Dr. Cliff Holloway, CEO of Scancell, commented, "It has been a strong period for Scancell as we continue to make significant progress with our pipeline of cancer immunotherapies. We have expanded our product opportunities through in-licensing and external collaborations, including with Cancer Research UK and BioNTech, who have provided further validation of our immunobody and immunotherapies platforms." The fundraisings this financial year have given us the necessary funds to progress our immunobody platform pipeline, and to continue to advance MODY1 towards the clinic. SCIB1 has demonstrated impressive survival data to date, and operational and regulatory activities are underway for the initiation of the international SCIB1 checkpoint inhibitor combination Phase 2 study in patients with melanoma. A full copy of the announcement can be found on the Scancell website, www.scancell.co.uk. This announcement contains inside information for the purposes of Article 7 of Regulation (EU) 596/2014 (SMAR). For further information, Scancell Holdings Plc. Dr. John Chiplin, Chairman 44-020-3727 1000. Dr. Cliff Holloway, CEO Panmure Gordon, UK, Limited, Nominated Advisor and Corporate Broker, Freddie Crossley, MRO 44-020 7886-2500, FTI Consulting Mo Noonan, Simon Conway 440-20-3727, 1000. About Scancell. Scancell is developing novel immunotherapies for the treatment of cancer based on its immunobody R and Monotope R technology platforms. Immunobody R vaccines target dendritic cells and stimulate both parts of the cellular immune system. They can be used as monotherapy or in combination with checkpoint inhibitors. This platform has the potential to enhance tumor destruction, prevent disease recurrence and extend survival. SCIB1, the lead program, is being developed for the treatment of melanoma. A Phase 1/2 clinical trial has so far successfully demonstrated survival data of more than 5 years. SCIB2 is being developed for the treatment of non-small cell lung cancer and other solid tumors. Scancell has entered into a clinical development partnership with Cancer Research UK for SCIB2. Monotope R represents a completely new class of potent and selective immunotherapy agents. It stimulates the production of killer CD4 T cells which overcome the immune suppression induced by tumors, allowing activated T cells to seek out and kill tumor cells that would otherwise be hidden from the immune system. Monotope R, alone, or in combination with other agents, has the potential to treat a wide variety of cancers. MODY-1 is being developed for the treatment of triple-negative breast cancer, ovarian cancer and sarcomas. For further details, please see our website, www.scancell.co.uk. Chairman's statement: I am pleased to report on the final results of Scancell Holdings Plc for the year ended the 30th of April 2018. During the financial year, Scancell has announced a number of exciting collaborations and agreements: a clinical development partnership with Cancer Research UK (CRUK) to fund and manage a Phase 1/2 study with Scancell's second immunobody R vaccine, SCIB2, in combination with a checkpoint inhibitor for the treatment of non-small cell lung cancer, NSCLC, was announced in December 2017. A research collaboration on Monotope R between Scancell and BioNTech to develop T-cell receptor (TCR) therapeutics was announced in January 2018. ESA Pharmaceuticals and Scancell entered into a collaboration agreement for the manufacturing, development and commercialization of MODY-1, AMPLIVA&T/R conjugates as announced in February 2018. The polypeptide group was contracted to manufacture Scancell's MODY-1, AMPLIVA&T/R conjugates as announced in April 2018. An agreement with the University of Nottingham to license several novel monoclonal antibodies against tumor-associated glycans was announced in April 2018. The company has also raised £11.6 million net of expenses from placings of shares at the beginning and end of the financial year. An additional £1.1 million net was raised post-period in May 2018 by an open offer to shareholders. These funds have and will enable the group to continue to progress the clinical development of its innovative cancer treatments. Since the end of the financial year, Scancell has announced the exercise of its option to license Ichor's electroporation delivery system, which enables Scancell to use the new TriGrid R v2.0 as the delivery system for its planned Phase 2 checkpoint inhibitor combination study with SCIB1 in patients with advanced melanoma. At the same time, Ichor exercised its option over 3,184,620 shares at 4.5p each. Immunobody R. Scancell's Immunobody R immunotherapy platform uses the body's immune system to identify, attack and destroy tumors. This is achieved by enhancing the uptake and presentation of cancer antigens to harness high-avidity T-cell responses. Each immunobody vaccine can be designed to target a particular cancer in a highly specific manner, offering the potential for enhanced efficacy and safety compared with more conventional approaches. SCIB1 Melanoma Vaccine SCIB1, Scancell's lead immunobody R, is in development for the treatment of melanoma. During the year, Scancell announced the publication of a peer-reviewed research paper on SCIB1 in the scientific journal Onco-Immunology entitled, Targeting GP100 and TRP2 with a DNA Vaccine, Incorporating T-Cell Epitopes with a Human IG1 Antibody Induces Potent T-Cell Responses that are Associated with Favorable Clinical Outcome in a Phase 1/2 Trial. The publication describes the outcome of the company's Phase 1/2 clinical trial of SCIB1 in patients with metastatic melanoma up to the date when all patients had received 5 doses of SCIB1 in the main part of the study. The paper concludes that SCIB1 is a novel class of anti-cancer immunotherapy that induces T-cells which can cause tumor regression in patients with melanoma. The high frequency of responses, their breadth and durability suggest that SCIB1 is worthy of further study in a larger cohort of patients. This is particularly the case in the adjuvant setting, where all of the patients responded immunologically and where absence of toxicity is an important clinical consideration. Furthermore, the stimulation of potent de novo immune responses by SCIB1 may provide an opportunity for synergistic combination therapy with checkpoint inhibitors in late-stage disease. The last patient treatment in the Phase 1/2 clinical trial occurred during February 2016. Scancell therefore ceased collecting ongoing survival data in February 2018, after each patient had been followed up for a minimum of 2 years. At that time, the final survival data was overall, 18 of 20 stage 3 IV melanoma patients with resected disease remained alive. Of the 16 resected patients who received 2 to 4 mg doses of SCIB1, only 6 patients had recurrence of their disease with only 2 deaths. All 14 surviving patients in this group had passed the 5-year time point since study entry. The 4 patients who had disease recurrence went on to receive other treatments for their melanoma. However, despite having received multiple interventions and recurrences prior to study entry, the other 10 patients had no treatment other than SCIB1. 1 patient with unresected disease also survived for more than 5 years since starting treatment with SCIB1 despite disease progression. *, 2 of 4 resected patients who received 8 mg doses of SCIB1 experienced disease recurrence although none had died. *, the median observation time for this group of patients was 35 months. *, all patients who relapsed went on to receive additional therapies for their melanoma. The proposed international clinical study of SCIB1 in combination with an immune checkpoint inhibitor is planned to commence by the end of this year subject to the necessary regulatory approvals. This Phase 2 study will utilize Ichor's new Trigrid V2.0 electroporation delivery device, and the company is currently in discussion with the U.S. Food and Drug Administration (FDA) regarding an investigational new drug application (IND). In parallel, the company is actively working on the necessary operational activities required to initiate clinical sites in the U.S. and UK. SCIB2 lung cancer vaccine. During the year, Scancell announced a clinical development partnership with CRUK to fund and manage a Phase 1/2 study with SCIB2, Scancell's second immunobody vaccine, in combination with a checkpoint inhibitor in patients with NSCLC. CRUK will be responsible for manufacturing the clinical trial supplies of SCIB2, conducting preclinical testing, and sponsoring and managing the clinical trial. Following completion of the trial, Scancell will have the option to license the rights to the data subject to paying an agreed fee and will undertake responsibility for further development of SCIB2. If Scancell does not exercise the option, CRUK retains the right to take the SCIB2 program forward in all indications, with any future revenues being equally shared. CRUK is an important partner with the resources, both monetary and clinical development expertise, to ensure the best chance of success in bringing the SCIB2 vaccine to patients as soon as possible, and also provides an important validation and endorsement of Scancell's immunobody platform. Patents. During the year, we also announced that a patent for Scancell's DNA immunobody technology has now been granted in Europe. the European patent, number 2,134,357, granted by the European Patent Office, covers Scancell's DNA immunobody (R) platform technology and is key to the protection of the company's pipeline of immunobody (R) vaccines, including lead candidates, SCIB1 and SCIB2. This key European patent further protects our global intellectual property portfolio. Moditope R. Scancell's Moditope R platform represents a new class of cancer immunotherapies based on stress-induced post-translational modifications, symptoms. Moditope vaccines are novel modified peptide-based immunotherapies developed to exploit both the process of autophagy, which occurs naturally in stressed or dying cells, and also the ability of CD4 cells to expose cancer cells for direct killing. This is achieved by stimulating the production of CD4 T cells using citrullinated tumor-associated peptide epitopes which overcome self-tolerance and destroy tumor cells. Clinical studies have shown unprecedented anti-tumor effects can be delivered without requiring checkpoint inhibition. MODI-1 is the first Moditope R vaccine and contains citrullinated peptides derived from vimentin and less than alpha greater than enolase. Vimentin and enolase peptides are highly expressed in triple-negative breast cancer, ovarian cancer and sarcoma. Preclinical data suggests that MODI-1 may be effective in up to 90% of patients with triple-negative breast cancer, up to 95% of patients with ovarian cancer and up to 100% of patients with sarcoma. The company announced in February 2018 that it has entered into a worldwide licensing and collaboration agreement to use ESA's AMPLIVANT-R adjuvant technology for the manufacturing, development and commercialization of MODI-1. Previous preclinical data demonstrated that conjugation of the MODI-1 peptides to AMPLIVANT-R enhances anti-tumor immune responses 10 to 100-fold and resulted in highly efficient tumor eradication, including protection against tumor re-challenge. This combination of MODI-1 with an enabling adjuvant technology such as AMPLIVANT-R has the potential to significantly enhance its efficacy in patients. The company subsequently announced the appointment of the Polypeptide Group, one of the world's largest independent contract manufacturers of therapeutic peptides, for good manufacture practice (GMP) compliant manufacture of the MODI-1, AMPLIVANT-R conjugates with the aim of filing a clinical trial application (CTA) in the UK and commencement of the planned Phase 1/2 clinical trial during 2019. MODI-2, the company's second Moditope program, MODI-2, is currently in preclinical development and is expected to address multiple cancer indications. MODI-2 will be the subject of new intellectual property applications with a view to extend the company's dominant patent position in relation to post-translational modifications of cellular proteins and their application in the treatment of cancer. Collaboration. Scancell has also announced that it has entered into a research collaboration with BioNTech for the potential development of innovative, TCR-based therapeutics for the treatment of cancer. This research collaboration combines Scancell's Moditope R immunotherapy platform and BioNTech's platform technology for high-throughput cloning and characterization of naturally selected T-cell receptors. Under the terms of the agreement, Scancell and BioNTech will enter into an initial research collaboration to discover and characterize T-cell receptors specific for citrullinated epitopes from vimentin and enolase. These epitopes form the basis of Scancell's first Moditope R development candidate, MODI-1. Upon completion of these studies, BioNTech will have the exclusive option to enter into a license agreement for the development of T-cell-based therapeutics that are specific to MODI-1 epitopes. Since the financial year-end, Scancell has extended its strategic research collaboration with the Rheumatology Unit at the Karolinska Institute, Sweden. The expanded agreement will explore the potential of the Moditope R platform to develop multiple immunotherapeutic agents for a range of different cancers. Scancell's research has shown that citrullinated proteins are involved in the control of tumor growth, and we believe that this expanded collaboration will help us to further develop Moditope R, not only for use in cancer vaccines, but also other cancer immunotherapy approaches including TCRs. Further endorsement of Scancell's Immunotherapies R platform resulted in the shortlisting of a proposal to meet CRUK's Grand Challenge in cancer vaccinology. This high-profile consortium, co-led by Scancell's CSO, Professor Lindy Dorrant, includes the proposed development of a new Immunotherapies R vaccine. The winner of the awards from the 10 shortlisted proposals is expected to be announced later this year. Patents. The European Patent Office has announced its intention to grant Scancell's application for a European patent for its Immunotherapies R immunotherapy platform. This patent is key to the protection of the company's pipeline of Immunotherapies R vaccines for the treatment of cancer and will provide commercial exclusivity in all major European territories including Austria, Belgium, Switzerland, Germany, Denmark, Spain, Finland, France, United Kingdom, Ireland, Italy, Netherlands, Norway, Poland, Portugal, Sweden and Turkey. Counterparts to this patent have been filed in Australia, Brazil, Canada, China, Hong Kong, Japan, South Korea, South Africa and the US. Monoclonal antibodies. Scancell has acquired, from the University of Nottingham, a number of novel monoclonal antibodies against tumor-associated glycans with the aim to further develop and identify lead therapeutic candidates. Alongside this, Scancell has also acquired a proprietary technology to enable the modification of the constant region (FC) of a human antibody to allow direct tumor killing. Monoclonal antibody therapeutics have proven to be effective in the treatment of many cancer indications, and identification of new products against novel targets are highly sought after in the field. Together these offer a complementary platform to Scancell's existing cancer immunotherapy platforms, ImmunoBody R and Immunotherapies R, and we look forward to informing you of further progress as we assimilate and advance these new assets. Financial profit and loss account. The group made an operating loss for the year to the 30th of April 2018 of £4,941,800 British pounds, 2017, loss of £4,548,836 British pounds. There has been a 3% increase in development expenditure to £2,855,264 British pounds, 2017, £2,766,098 British pounds, and a 17% increase in administrative expenditure to £2,086,536 British pounds, 2017, £1,782,738 British pounds. The rise in administration expenses is due to an increase in licensing and patent costs for both the ImmunoBody R and Immunotherapies R platforms. Overall, the loss for the year was £4,194,509 British pounds, 2017, loss £3,544,979 British pounds. Balance sheet. The cash at bank at the 30th of April 2018 was £10,303,168 British pounds, the 30th of April 2017, £2,672,335 British pounds, and net assets amounted to £13,940,950 British pounds, the 30th of April 2017, £6,499,325 British pounds. Share capital. Placing on the 11th of May 2017, the company placed 50,499,999 ordinary 0.1p shares at a price of 10p per share and raised 4.7 million British pounds net of costs. A further placing of 62,411,000 ordinary 0.1p shares at a price of 12p per share, on the 20th of April 2018, raised 6.9 million British pounds net of costs. Since the year-end, there has been an open offer to shareholders that raised 1.1 million British pounds, net of expenses, and Icor exercised a tranche of their share options, raising a further 143,307 British pounds. Directors. At the last AGM in October 2017, we announced the appointment of Dr. Cliff Holloway as Chief Executive Officer of Scancell, and Cliff took up his position on the 10th of January 2018. Since the year-end, Kate Cornish-Foden stepped down as a non-executive director of Scancell on the 31st of August 2018. Kate has made an important contribution during her time on the board and in her role as chairman of the Remuneration Committee. I would like to thank her for her hard work and wish her all the best for the future. staff. The board recognizes that the progress made over the year would not have been possible without the dedication and support of all our staff, and, on behalf of the directors, I offer our thanks to them. Outlook: the company continues to make significant progress with our pipeline of products, SCIB1, SCIB2, MODY1 and MODY2, as well as expanding our product opportunities through in-licensing, anti-glycan antibodies, and external collaborations. Fintech TCR Research Project. Operational and regulatory activities are underway in the US and UK for the planned initiation of the SCIB1 checkpoint inhibitor combination Phase 2 study in patients with melanoma, and the new funds raised at the beginning and end of the financial year have provided Scancell with the cash to progress both this trial, and the continued development of MODY1 towards the clinic. The data generated from these clinical studies, if positive, will allow the company to create new value which can subsequently be realized through negotiation of commercial transactions. To maintain this trajectory, the company will continue to explore additional funding options to ensure that the development programs continues to be properly resourced. John Chiplin, Chairman, Consolidated Profit or Loss and Other Comprehensive Income Statement 2018-2017 for the year ended the 30th of April 2018. British pounds GBP. Development expenses £2,855,264, £2,766,098. Administrative expenses £2,086,536, £1,782,738. Operating loss, Note 2, $4,941,800, $4,548,836. Interest receivable and similar income $2,753, $53,445. Loss before taxation $4,939,047, $4,495,391. Taxation, Note 3, $744,538, $950,412. Loss and total comprehensive income for the year $4,194,509, $3,544,979. Earnings per ordinary share, pence. Note 4. Continuing operations: basic 1.34p, 1.36p, diluted 1.34p, 1.36p. Consolidated statement of changes in equity for the year ended the 30th of April 2018. Share retained capital premium option earnings total GBP balance the 1st of May 2016 $261,558 $21,785,295 $649,652 $12,704,224 $9,992,281. Loss for the year $3,544,979 $3,544,979. Share option charge $52,023 $52,023. Balance the 30th of April 2017 $261,558 $21,785,295 $701,675 $16,249,203 $6,499,325. Share issue 112,911 $12,426,409 $12,539,320. Expenses of issue $837,080 $837,080. Loss for the year $4,194,509 $4,194,509. Share option charge $66,106 $66,106. Balance the 30th of April 2018 $374,469 $33,374,624 $635,000.